Hello, my name is James Dries and I'm completing a Bachelor of Exercise and Sports Science at the University of New England. I'm completing this presentation as a part of an assignment for EXSC 335, Motor Control and Human Development, which is coordinated by Dr. Dominic McNeil. The topic for my presentation is based upon a laboratory activity relating to the central nervous system and motor performance. As we all know, a major aim of sports scientists, strength and conditioning coaches, PE teachers and sports coaches is to develop the motor skills of athletes and students in order to gain a better performance. Learning and motor control are different yet related concepts. Before we describe the details of the recently conducted lab activity, I'll first describe the concepts of learning and performance, and I will also be discussing the concept of motor control and its relationship to performance. I'll then proceed to explain the methods and the results of the lab activity before discussing the proposed neurological mechanisms of how learning has occurred. Okay, let's start off by addressing the concepts of learning and performance. So, learning, it refers to the changes in the capability of a person to perform a skill. It's an internal process and can't really be measured directly. Therefore, to see if learning has occurred, it must be inferred by measuring performance. So performance, well, it's an observable behaviour of a skill and the skill's execution at a specific time and in any specific situation. Performance can be measured because it's observable. So what is motor control? Well, motor control is our ability to direct and regulate the essential mechanisms of movement. It addresses such topics as the role of the central nervous system in muscle and joint coordination that facilitate functional movements, how we use sensory information from the environment to modify movements, and how the environment influences our behaviour. Well, the aim of this lab activity was to examine the performance changes associated with learning and how these changes may be facilitated through activation of specific neural locations. Well, for this lab activity, we had 33 participants completing a pencil maze task while they were blindfolded for 15 trials per person. Each trial was completed with the intention to do it as fast as possible, and the time taken to complete each trial was recorded. Penalties were assigned each time the correct path was deviated from. Participants weren't allowed to see the maze prior to or in between trials. And the equipment that we used was a plastic pencil maze, paper for the maze to be placed on, a pen or a pencil, and a blindfold. Here we have a maze trial example from one of the participants after they completed their 15 trials. In the bottom right corner, you'll see a yellow circled area, and from this point on, the correct path follows the yellow highlighted line. Everywhere else is a deviation off the correct path, and you'll see that the bits that are coloured in like a darker blue, that's where people were getting stuck and probably having issues or problems with finding their way through. Here we have a graph of the raw data of the average time taken to complete each trial. There was a few outliers in the raw data, so I've removed those to give a better representation of the learning curve and to also ensure that they weren't skewing the data. Here we have a modified version of the graph with outliers removed. And you'll see the curve is a negatively accelerating or decelerating curve. And on the x-axis, trials are representative of time. And on the y-axis, we have performance measured in seconds. And the relationship here is that as time passes or trials occur, the performance is increasing because the time taken to do each trial is decreasing. Now, initially it took a lot longer to complete the first few trials, but performance started to improve after those trials until towards the end where the performance curve almost plateaus, indicating that improvement had occurred and consistency with motor performance was increasing over time. Now the first trial, it took over 3 minutes, and the second trial, the average time was almost 2 minutes. So that's a 101 second improvement from trial 1 to 2. And from trial 2 to 3, there was a 16-second improvement. And trial 3 to 4, there was an improvement but only 7 seconds. And from trials 6 through to 15, improvement occurred but at a much lower rate. Now, because this vast improvement was occurring over a short time frame, this indicates that the skill level required to do the maze task was low. And this can be compared to the amount of hours of practice required for an elite baseball pitcher to reach their level where the skill level required for them is quite high. So how does all this happen in the body? And what is happening in the body to allow for this to happen and relay all this information? Well, we're using feedback from receptors in our hand, and this is known as somatosensory input. We get sensory input from cutaneous mechanoreceptors in the hand, as well as proprioceptors, which also supply information about the hand position relative to the maze puzzle and feeling the edges of the maze because we can't see anything. And this sensory information then travels along afferent neurons that synapse with the interneurons in the spinal cord, which then passes the information along ascending tracks to the brainstem. This information from the hand regarding its position and details of the maze are then relayed from the brainstem to the cerebellum and the thalamus, which then relays the information up to the somatosensory association area in the parietal lobe. Now, in this parietal association cortex, this is where the assessment and interpretation of the sensation involved in positioning and manipulating the hand to complete the maze task occurs. Once information processing is complete, we can then start to initiate the movement. And this really begins in the somatosensory cortex. Here a response plan is developed to the incoming stimulus, such as, what will I do now that I've hit a dead end in the maze? And a response plan to that stimulus is then passed onto the premotor area for advanced planning and coordination of an intended movement, such as, you've hit the dead end of the maze. Obviously now we can't keep going that way, so let's go back and look for another option. And it also receives information from the thalamus and the cerebellum and passes the advanced plan onto the SMA. Preparation of the intended movement response occurs in the supplementary motor area, or SMA, and it's associated with conceptual ideas. The SMA uses memory of previous attempts or similar movements to prepare upcoming movements, then modifies the movement execution, and it receives additional input from the thalamus and the basal ganglia. Well, now it's time to make a movement, and this is where the M1 comes in, and it's responsible for the execution of the intended movement, and it also controls the parameters of the required movement. Information is passed from the M1 along descending pyramidal tracks to efferent motor neurons, which generate muscular contractions to execute the movement. This information is also passed along extrapyramidal tracks to influence postural control and to maintain a stable body for the task to be completed. Let's now address how performance modifications occurred during each trial. We've got two areas involved here, and the first is the cerebellum, and it acts as a comparator between the intended movement and the actual movement. It's kind of like an error detector and corrector, and it sends output to the primary motor area and the M1 so that the movement can be modified as necessary. And its main roles are in modifying externally generated movements, such as like in the response to an incoming stimulus. And it's linked to visually triggered and guided movements, which kind of weren't really the case here because we were blindfolded. Now, the other area is the basal ganglia, and it's a relay station, and it receives information from the motor areas and sends that information back to the motor areas via the thalamus, and then sends the motor commands through the brainstem onto motor units. And its role is to scale and modify parameters of movement. And it's responsible for internally generated movements such as cognitive processes, and this is more so involved in making a choice and less from reacting to a stimulus. Now that we've covered the sensory input and motor control of performance, let's examine the physiological processes involved in learning and performance improvement. Well, this occurs via neuroplasticity, and it's the ability of the nervous system to undergo persistent or lasting modifications to its structure and function. Physiological changes occur at neural junctions, and at neural junctions we've got neural end plates, and they can increase in diameter, and we can also get neurotransmitter and neurotransmitter enzyme activity altered. Neurons also branch and form other connections with nearby neurons. And neuronal growth and branching with the hippocampus within the brain also contributes to spatial learning and memory. We also had memory involved in performance improvement because details of past performances aid future performances. And this is associated with the limbic system, and it's involved in facilitating memory storage and retrieval, while memory is also stored in the frontal lobe of the cerebral cortex. Part of the limbic system is the diencephalon, and it's a system that integrates sensory information within motor output and is able to use memory as a reference. And the type of memory we used in the maze task is procedural memory, and it's the memory of how to do something. As a participant completed each trial, characteristics of the maze were stored and used for the next trial. To summarise, as we could see in the results, performance improved over time, and this was due to neuroplasticity enhancing memory and motor control. We could then use the details of our past performance to modify present and future performance in order to change movement characteristics and parameters. Well, hopefully this presentation has demonstrated how motor performance is related to the central nervous system and how if we want to determine if learning has occurred, we have to assess performance. This is quite often the reason we sit exams. As you may now appreciate, there are many parts of the nervous system involved in motor control, and the execution of movement requires a lot of processing in a short period of time. So as neuroplasticity occurs, we get physiological changes in the nervous system that allow for improved performance. It's not just a matter of remembering how to perform a task, but our nervous system also adapts. It was noticed that after the trials were completed, a few participants attempted to do the maze as quickly as possible without a blindfold and found that they were reverting to the pattern they had developed while blindfolded and still making some of the same errors. This would suggest that a motor program for the maze had been stored within the CNS and lends support to the notion of learning a task correctly in the first place. Otherwise, retraining is required to relearn the movement or skill and break bad habits.